Since I'm sorry I only could make it this afternoon, I looked at the program for the other days, and it all looked very interesting. The whole setting is very interesting. I think physiology and imaging is something that really makes me happy anyway. So I'm glad to be here. I will not talk much about MRI because I don't know much about it. There will be some talks about transcranial Doppler. I have no disclosures, but I am editor at JCBFM. And when I looked at participants at the meeting, I saw some familiar names, people on the editorial board, people I sometimes ask to review papers. So it may be good to know that we are looking at this topic for the journal. Brain physiology is very interesting, especially if there are any clinical links. So please send us your best papers, but also help me review the and make sure we select the high quality papers for that journal. So just to make sure, I hope this is all basic knowledge for you. The brain gets a tremendous amount of blood flow. It has a really absurd amount of blood vessels going into it. 3 main vessels, 2 carotids, and a vertebral artery that are way too large for the size of the organ. It has this big roundabout, a circle of Wills. And then an important aspect, I think, is that the brain is perfused from the outside in, which is in contrast to all other organs we have, which are perfused from the inside outwards. It's most of them. What I find in many studies that do brain physiology is that they tend to lump all regulation processes together. For example, everything is called either autoregulation or brain reactivity or phasal reactivity. Whereas in fact, there are quite a few really succinct processes in the brain going on that you cannot just lump together. And this lumping together leads to, it's not just semantics, it really leads to errors in interpretation, but it also has some clinical implementation, which I'll talk to you about. 2 years ago with these colleagues, we wrote a huge, sort of, it got out of hand, review paper. The ones in physiological review tend to be really huge. I think, remember, this one had about 600 references. It was quite an honor, I found, to write. It was the follow-up paper to Niels Lassen's autoregulation paper in Physiological Reviews in 1959. So we tried to capture all the new information on autoregulation that has been obtained since 1950. What we try to emphasize is that there is a lot of processes that are involved in brain blood flow regulation. And autoregulation is one of them, but it's a distinct phenomenon. And what we like to do is to limit the concept of autoregulation to pressure autoregulation, which is also the way it's been handled in the rest of the body, for example, in the kidney and other organs, where autoregulation is meant to be how how blood vessels, or let's say the organ, responds to changes in pressure. And I'll come back to that in a bit. So important to realize, I think, as I said, the brain is perfused from the outside in, which leads to the fact that in most parts of the brain, the parenchyma, there is this you have the surface brain vessels, and then there is a perpendicular entrance of vessels into the brain. This does affect the perfusion pressure of the tissue, and then there is tremendous branching of these vessels into arterioles, which affects the perfusion pressure of the brain. And as you will see, that there are some exemptions. For example, the hippocampus is perfused from the posterior circulation, which has much less branching before it reaches the hippocampus. So in terms of the effects of pressure changes on brain tissue that we had in the talk before, I think it could be that, for example, the effects of blood pressure on the hippocampus could be much larger than the pressure effects that you would have down here. It's good to realize that those differences exist. Another thing that's important is that there is this question about where, which parts of the vascular bed contribute most to vascular resistance. So we had a bit of a discussion on resistance in capillaries, for example. What I believe from a physiological standpoint is that most resistance is created by the arterioles. They have the huge vascular surface. There's not that much contribution from the what we call the conduit vessels, so the carotid artery and the MCA. They can constrict and dilate a little bit, but the total outcome on vascular resistance is relatively small. So most is achieved by dilation or constriction of arterioles. Now, there is much discussion on what the role of capillaries is in brain resistance. I'm not an expert. The idea is that something that they only dilate passively. Others say, well, maybe parasites or astrocytes can really affect capillary diameter, and in that way they may contribute to vascular resistance. And also, recent research has sort of suggested that it could be that, for example, in inflammation, where there is adhesion of leukocytes that may obstruct capillaries, that would really affect brain blood flow at a very downstream vessel. So when I sometimes look at global brain blood flow and I'm thinking about autoregulation and how the brain responds to brain pressure changes, the blood pressure changes, we should not forget that there could be situations where there is, for example, inflammation and leukocyte stallment in capillaries, where there may be areas of the brain that are starved from perfusion, while global perfusion actually should be. So it's a complex system. And what I try to do is simplify it by separating it in three distinct areas. So there is cerebral autoregulation, which is how blood vessels respond to blood pressure. There is CO2 reactivity, or a cerebral vasomotor reactivity to carbon dioxide. And there is neurovascular coupling. And what I said in the beginning, in many papers, all these things are lumped together and called reactivity or called autoregulation. But that's confusing, I think, because there are really distinct mechanisms. For example, neurovascular coupling is the response to neural activation. It may be linked to metabolic activity and maybe to CO2, but there is also quite some evidence that there is a direct downstream influence of neurons on upstream capillaries, for example. And you cannot simply lump this together with CO2 reactivity. And these two are completely different from pressure reactivity. So that's something to keep in mind during my talk. And I'll explain a bit later why this is more than semantics. But I'll talk about cerebral autoregulation only. This is Lassen's curve from 1959. So the paper we updated 2 years ago. It's a well-known concept where you have this plateau phase where brain blood flow is thought to be held relatively constant. And it's a very wide range of blood pressures. These are mean arterial pressures. So the ones here where where CBF starts going down is really the type of blood pressure where people are in septic shock, right? It's not healthy. You're not supposed to be here. The same goes for here, the upper limit, 150, that's really, really hypertension. It's not just average hypertension, it's really the hypertension that is going to get you killed. So beyond these extreme levels, CBF is held stable. This is the classic concept. The first question is this really true? And it has been debated in the last decades whether this concept is really true. And I will explain to you why. Now, this is the actual curve that Niels Lassen produced in 1959. It's only part of the curve. The upper part was added later on. What he did is he looked at a number of groups of people, patients really. So these numbers are groups of people with certain hypertensive disorders. And what he found is that regardless of their average blood pressure for the whole group, their CBF levels, which were measured at that time by xenon SPECT, were almost the same, more or less the same. So his conclusion was that there is an autoregulatory process in the brain that keeps brain blood flow constant, despite a quite large range of pressures. There was one group, only one group, that had really low blood pressure and had low CBF. So he concluded, and I think there's not much debate, that beyond a certain level, this autoregulation doesn't work and you get hypoperfusion. But this is what he measured. Now it's really difficult to validate this in humans, in individual human beings. You cannot pull a normal human being through the whole autoregulation curve. You cannot increase somebody's blood pressure to 200 over 100 systolic and then reduce it to 70 over 50. It's physiologically not possible. It's also ethically not possible. You won't get any ethical approval to do that. You can do it in animals. It has been done in dogs, rabbits, cats, baboon, mouse, rat. And you do see a sort of approximation of Lassen's curve. There is a plateau phase. And beyond that plateau phase, there is an increase or a decrease. There's much to say about these studies. And one thing you may notice is that the plateau is much less wide as in the Lassen curve. That has to do with the fact that it's even in these animals, it's really difficult to create all these different levels of pressure and keep them stable. It's almost impossible. But anyway, the concept has been validated. And it's now called static autoregulation. And I'll explain to you why it's called static autoregulation. Within this plateau phase, it's not held exactly stable, but relatively stable. It can still go up and down with about 10%. It is valid for a wide range of blood pressures, but the key element is that it's only applicable for really slow changes in pressure. It's called static autoregulation because it applies to a more or less steady state situation where blood pressure has changed really slowly. And the best way to think of it is in weeks. So for example, somebody went from having high blood pressure to normal blood pressure by getting treatment over 6 weeks, for example. In those cases, if you measure somebody's cerebral blood flow, it will be exactly the same in both conditions. And what we think is that the main regulatory mechanism is relatively simple. It's the vascular smooth muscle. So one thing you read in almost all papers on autoregulation is that it says it's a complex mechanism that is a combination of neural, myogenic, and metabolic processes, which is incorrect because that's a description of all processes together. That's a description of CO2 reactivity, neurovascular coupling, and autoregulation. Autoregulation in itself, pressure autoregulation, is really simply the Baylis mechanism, and it's related to smooth muscle. I will not go much more into hardcore physiology than the next two slides. What happens is that if there is a pressure response, which you see here, there is an acute diameter change that is brought about by vascular smooth muscle. This situation, you see that if there is a model where you have calcium present, you will see that if there is an increase in pressure, there is a limited initial increase in diameter because there's more pressure and the vessel will dilate. But then the Baylis effect becomes active. There is active restriction and the diameter will decrease. If you block this vascular smooth muscle and there is no calcium present, there will just be passive dilation. And this is what we now think. There's much debate on which mechanisms actually are responsible for sensing pressure in the vascular smooth muscle. This is the most recent review I could find also in Physiological Reviews by Davis et al. If you want to know more about this, about how these processes work. So it's well, if you say in physiology it's simple, it's never simple, but it's a relatively robust mechanism. It's smooth muscle that responds to pressure. And we have lots of vessels that, lots of materials that have smooth muscle. And apparently it takes quite a bit before the system gets damaged. So that's a reassuring. Now we now call it static autoregulation because since the 1980s, we have discovered a new area of autoregulation called dynamic autoregulation. And what made it possible is the sort of the discovery or application of transcranial Doppler, which that has the potential to measure brain blood flow at a really high temporal resolution, much higher than MRI, although MRI is getting there, which will make it more interesting for you guys. What transcranial Doppler does, for those who don't know it, it uses ultrasounds to detect the velocity of red blood cells in the middle cerebral artery, for example. You see these waveforms here that measure systolic and diastolic velocity. Now, under the assumption that the diameter of the middle cerebral artery doesn't change, then the changes in velocity are proportional to changes in flow, which means that you can deduce changes in flow, so not absolutely flow, by measuring Doppler ultrasound. We combine that with something we call VenaPress, which is a device that measures arterial blood pressure at the finger with every heartbeat. And so if you combine those, you can measure with every heartbeat, or actually within the heartbeat, systolic, diastolic, you can measure changes in blood pressure and changes in brain blood flow, or actually more correctly, changes in velocity proportional to flow. And what you will see, this is why we call it dynamic. This is the physiological response to standing up. Standing up is something you cannot measure in the MRI. It's also why you need DCD for these experiments. If you stand up, your blood pressure drops and it recovers. It's called an orthostatic blood pressure response. It's because your blood volume moves from your thorax to the abdomen and legs. That really leads to a reduction in pressure. As you can see, that leads to a transient reduction in brain blood flow, and it recovers. If you look really closely, you will see that brain blood flow recovers faster than pressure here, which is an element of autoregulation. More simplified here. And then a simple way to analyze whether dynamic autoregulation works or not is this. So this is the, you can do an experiment where you have somebody stand up, for example. You measure their blood pressure, and you measure the response of cerebral blood flow. If there is no autoregulation, then CBF would just passively follow the drop and recovery in pressure, which is this line here. If there is autoregulation, initially it follows the drop in pressure because it takes some time for the drop in pressure to activate your smooth muscle and for them to dilate. This takes around 5 seconds. There's always a 5-second delay in the response. Then they start dilating and CBF recovers back to baseline. Now, what doesn't happen is this dotted area. CBF is not held stable. That is one of the discussion points we've had quite a while. Based on Blossom's curve initially, many people thought, and many people still think, that CBF is held constant at all times. But even if there are transient drops in blood pressure, that CBF wouldn't change, but that's not true. So even if autoregulation works normally, it will change if the change in blood pressure is faster. And now, Wolf Diel in Germany, actually the first to try to quantify autoregulation. And you're all physicists, so I can talk to you about phase shifts, I think. What he did is ask people to breathe in a certain rhythm. We've talked about breathing patterns when I arrived here. If you do paced breathing, at 0.1 hertz, which is 10 seconds in, 10 seconds out. Due to the changes in thoracic pressure, your blood pressure will also start changing. So we talked about how breathing affects your physiology. Well, here you see an example. If you do this spaced breathing, you will get a 0.1 oscillation in arterial blood pressure. And as I told you, because this is relatively fast, this is 10 seconds, CBF will be affected. It will not be exactly stable. It will go up and down. But what you will see is that the oscillations have been shifted to the left, and the phase shift is around 60 degrees. Now, it's always difficult to understand why it shifts leftward, because it almost seems that the changes in flow are anticipating the changes in pressure. That's some homework for you to do tonight. Try to figure that out, why it shifts leftwards. If there is a phase shift, then you have normal autoregulation. If it would follow blood pressure passively, there would be it would be synchronized and it would be no phase shift. So phase shift is one way to quantify dynamic cerebral autoregulation, which is explained here. So it shifts 60 degrees. The second way is to quantify damping. And damping just means that the amount of change in pressure, and you compare that to the amount of change in flow. It's a simplified. Those are the two parameters we use to quantify dynamic autoregulation. Now, what have we learned? We found out that if you measure autoregulation this way, the effectivity of autoregulation really depends on how fast your blood pressure changes. So if it's really very slow, hours or days, and you get static CA and then it's really effective, then CBF doesn't change unless you get really low or really high pressures. But the faster the changes in pressure become, the less effective autoregulation becomes. And if it's very fast, then there is no, well, autoregulation is there in theory, but it doesn't have time. So this is how you could graph that in theory. If you have a really slow, even large increase in pressure, let's say over hours or days, you'll see that within certain limits, it will only mildly affect CBF. But if you have this same change in pressure happening in seconds, CBF will change in parallel. So it means that really fast changes in pressure can lead to hyperperfusion and to damage to your brain or to severe hypoperfusion. And again, something that we experience in daily life, I think, is when you get up, this is younger adults, older adults, this is pressure. When you stand up, there is a drop in pressure and it recovers. And it will affect your brain blood flow. And this is why if you've been sitting like now and you stand up quickly, you will feel a little bit dizzy. And it recovers. So no worries. Now we've applied this concept to clinical medicine. See what happens to autoregulation in hypertension, in aging, and in stroke, and in Alzheimer's. I started out my research in Alzheimer's disease. There hadn't been any studies at that time when I started, which was in 2004, I think, on autoregulation in Alzheimer's disease. And our hypotheses were that it would be impaired, because what you see in Alzheimer's disease is that these people have small vessel disease. They have modeling atherosclerosis, vascular stenosis. They have endothelial dysfunction. They have inflammatory processes. They have amyloid angiopathy, and they have blood-brain barrier dysfunction. So almost everything you can imagine in your brain blood vessels is affected in Alzheimer's disease. Why is it important to know about autoregulation in Alzheimer's? Well, one is that if you assume that autoregulation is impaired, the idea would be to maintain blood pressure at a higher level, because the fear is that if you lower it too much and they have no autoregulation, you cause hyperperfusion. And this has led to clinical advice to maintain higher blood pressure in older people, for example, and to not treat hypertension. Specifically not in people with Alzheimer's, but also in older people with small vessel disease. Drew some pictures of how small vessel disease looks like. One of the ideas is that in hypertension, the autoregulation curve would be shifted to the right to maintain, to withstand the high pressure. But then if you would lower blood pressure, then the lower limit of autoregulation would also move upwards. It would cause hyperperfusion and cause brain injury. Now, we have looked at a lot of studies, including studies that have used MRI in people with hypertension who were measured at their CBF measured before they were treated and then after. The study above here is done with transcranial Doppler. These studies are done with MRI, phase contrast, and arterial spin labeling. What you will see is that despite the large reductions in pressure, there is no drop in CBF in these patients. Now, these are older hypertensive patients without cognitive impairment. But the whole theory that the rightward shifted autoregulation curve just simply is not true. You treat hypertension, brain blood flow will remain stable. So that's clinically relevant finding, which we published in Hypertension a few years ago. In fact, it seems that, especially in the older people, if you treat hypertension, CBF increases, which is something you see in quite a lot of studies. Sometimes it reaches significance, sometimes borderline. But in all these studies, you see a trend towards an increase in CBF if you treat high blood pressure. So something to keep in mind, but it's completely in contrast to the hypothesis that there was that CBF would be reduced if you lower blood pressure in older people. Now, we've looked at whether autoregulation is impaired with aging. This is also something you read in many textbooks. We do know that if you look at aging, global CBF is reduced by around 0.3% per year in normal aging. So there is a gradual decline in CBF. But the process of autoregulation is perfectly maintained with aging. It doesn't matter what type of technique you use, whether it's Doppler, MRI, or PET scan, and whether you look at static or dynamic autoregulation. Aging does not affect autoregulation. Our most recent study has been done in people who were 70 originally, participated in a study we did in our lab, and then we re-invited them 10 years later when they turned 80, and we measured their autoregulation again, and it was unchanged. So that's good to know, but still it's in many textbooks. We looked at what happens to autoregulation in hypertension. So it's often assumed that the process of hypertension, as I said, shifts the autoregulation curve rightwards, and it affects autoregulation, and people would become more sensitive to hyperperfusion. Actually, if you look at the literature, it's been known since almost the 1970s, I think. All the studies that have been done do not find any impairment in autoregulation in hypertension, dynamic or static. Again, that is important because it does affect clinical guidelines, which still to date say that if you are, especially in older people, you have to maintain higher levels of blood pressure than in younger people. And they often cite as the reason for that autoregulation is impaired and you may risk hyperperfusion. The only thing, it's not true. Right? And the final thing, Alzheimer's. This is where I started my research, and we were really convinced that they would have an impairment in autoregulation. Alzheimer's patients have progressive reduction in global CBF by about 3% per year. So that's 10 times more than in normal aging. They have amyloid angiopathy. They have often have white matter injuries, macular infarctions. Some think they have autonomic dysfunction. It's not really sure, but anyway, many reasons for them to have impaired autoregulation. But we haven't found it. The first study I did was with Ron Zhang in Dallas. We measured, I think, 12 Alzheimer's patients. We were convinced to find impaired autoregulation. We didn't. We didn't really trust our results, so we repeated it in 20 people, and we still didn't find any impairment in autoregulation. And then we repeated it in 50, and we still didn't find any impairment in autoregulation. So by that time, we were convinced that it's not impaired. Some of the work has been done with transcranial Doppler. The last work we did, we combined it with arterial spin labeling MRI. Because people started mistrusting our TCD results. But this citation here from Sasulia and Powers in 2010 was done with PET. So it's not just our lab that finds it, it's all the few people who have looked at autoregulation in Alzheimer's all find that it's unimpaired. Now it may confuse you because what is impaired in hypertension and in some aging and in Alzheimer's is CO2 reactivity. CO2 reactivity goes down with vascular disease. It's severely impaired in Alzheimer's. But that's true. And neurovascular coupling is affected in hypertension and probably in Alzheimer's disease. But autoregulation, if you clearly define it as the response to is not. And why am I stressing that? It's not just semantics, it's because it affects clinical treatment. A lot of studies that I've seen published that say autoregulation is impaired in Alzheimer's. If you look at it, they've really measured CO2 reactivity or neurovascular coupling. But they are used in clinical guidelines to say, well, we probably should not treat hypertension because autoregulation. So that is why it affects clinical medicine. So that's why I sort of stress to keep the definition straight and separate autoregulation from the other processes. The final bit, and then I'll stop. Something that's interesting, I think, if you're in an MRI, you're always lying down. There is no hydrostatic gradient between heart and brain. But as soon as people stand up, there is. And there has been a huge debate in physiology on whether or not there is a hydrostatic gradient between the heart and brain. Well, obviously there is. Right? If you're supine, this is your heart, this is your head. Your mean perfusion pressure, let's say, is around 95, and it's the same in your heart and in your head and in your feet. And perfusion pressure is the difference between arterial pressure and venous pressure. Now, the difference between arterial pressure and venous pressure is more or less the same across the body. So this is your perfusion tissue perfusion pressure. Right? Not much exciting going on. But if you stand up, gravity starts coming into place, and there is a pressure reduction between heart and head level of. This is called the hydrostatic gradient, so 50 centimeters of water, which is around 37 millimeters of mercury, which means that if your systolic pressure is 120, that means that your pressure at heart level is around 90 systolic. Now, the debate has always been, does this affect brain perfusion? Is the brain gradient perfusion much lower in the head when you're standing than when you're lying down? But some assume that for the rest of the body, it's relatively simple. If you look at this graph, if you stand up, the hydrostatic gradient to your feet works the other way around. It gets around 95 millimeters of mercury added thanks to gravity. So the arterial pressure in your feet is 200. But your venous pressure also goes up. It has the same hydrostatic gradient. So that means the difference between arterial and venous pressure remains more or less the same as when you're lying down. That is that explains why your feet don't explode when you stand up. So it's really simple. Now, if this should also apply to your brain, that would mean that you would have to have a negative venous pressure in your brain. And there's been lots of debates on whether that's physiologically possible or not to have a negative venous pressure. Right? So the debate, which you will still see in many studies, is whether studies that you do, for example, in the MRI, when people are supine, and you find, for example, certain relationships between pressure and brain blood flow, whether they still apply to daily life when people are not supine all the time. They sit up, they stand up. Nobody can really measure it. But we came up with a good excuse to do parabolic flights. I was inspired by this. Like I said, my first research I did with Rong Zhang in Dallas. They worked together with NASA. And I found out that many exercise physiologists tend to work with space medicine. And in Europe, it's the European Space Agency. And we had a collaboration with an exercise physiology group in Cologne. And I managed to get myself into one of these programs. And really just for fun, to be honest. But I invented a reason. Now, I don't know if you're familiar with parabolic flights. What they do is they fly. It's a commercial plane. It's a really old plane. In the 1980s. It just flies. It flies over the ocean because if it crashes, then it will only kill a few fish. So they fly from Bordeaux airport. Okay, so they have two oceans nearby. What they do is they pull up steeply, where your gravity increases from 1g to about 1.8. Then it enters the parabola, where there is sort of a free fall. There is zero gravity, well, zero experienced gravity during around 20 seconds. Then it has to pull up, where you get an increase a sudden increase to 1.8 G. So you go from normal to double G to zero, and then from zero to double. It's quite heavy. We all looked really happy before we boarding the plane, especially me. This is us preparing. So it's an old spacecraft they bought from the German government. Angela Merkel flew in this one, and they padded it, and they have all sorts of labs on the floor. This was our lab, where there is a screen, a transcranial Doppler, and a finger press device, and some EEG devices. We start early in the morning. This is during a parabola where you see people flying around. These guys here are the stewardesses or stewards, and their job is to make sure people don't float over experimental tables, because when the plane pulls up, they fall down. It happened to me. I got lost and I started floating, and they had to rescue me. They all look really happy. And this is all the experimental setups. So people do human physiology, but there are also plant experiments and experiments on how cement hardens without gravity. It's an interesting bunch. And if your experiment is done, then you can get to play in the play area. It's weightlessness. This is where I got the grant from. This is the serious stuff. What we did is we measured brain blood flow using transcranial Doppler, pressure with the finger press, and heart rate. And what you will see is that this is just baseline. This is during the pull up, the 1.8 G. You see a slight increase in pressure at that time. This is 0 G, weightlessness, where there is a huge drop in pressure, and then a sudden increase in pressure again when there is 1.8 G. Now, this is a whole different area, the physiology behind explaining the blood pressure shifts. That's because of the distribution of thoracic abdominal fluid by gravity. But anyway, it's good to know that these large changes in pressure are there. What you see in CBF is these strange increases here, a decrease here, an increase here. So what we did is this is blood pressure. It doesn't say anything about pressure at heart level. Now, if you calculate blood pressure at brain level by measuring the hydrostatic gradient between heart and brain, and then correcting blood pressure for that. So the hydrostatic gradient is, on average, is 30, which is the difference between heart and brain during 1G. Now it almost doubles, of course, 5 minutes. Yeah, oh yeah, I should make it. It doubles when gravity doubles, right? Because the hydrostatic effect of this column of water doubles if your gravity doubles. So it goes from 30 to around 60. In 0G, there is no hydrostatic gradient. There is no gravity. And then here it goes from 0 to 60. And if you correct your blood pressure for this hydrostatic gradient, you see that during weightlessness, there is this sudden increase in pressure. And it recovers. And then when you go from weightlessness to 2G, there is this sudden drop in pressure. And this explains these sudden increases here and here in CBF. So what we think these experiments tell us is that there is an effect of hydrostatic gradient on brain perfusion. Now, why is that clinically relevant? I had to figure that out because otherwise I wouldn't be able to get my research reimbursed. It does matter if you, the way, for example, you position your patients does matter. If somebody is hypotensive, for example, you should not set them upright because that will cause another reduction in their brain perfusion and will lower their CBF. Vice versa, if somebody has a hypertensive crisis, you should not put them flat down, but have them sit up. These simple things really help our understanding. And there have been quite some debates with people who said, well, the hydrostatic gradient doesn't apply to the brain. Said, well, it doesn't really matter how you position a patient, whether you put them head down or head up, it doesn't affect their brain blood flow. So anyway, it does. Of course, this is not definitive proof, but it was certainly fun to do. I will leave it at that. If you want to read more about regulatory processes in the brain and you have a few weeks, then I recommend it. Thanks a lot for your attention.